All right, class, here in chapter 13, we're going to talk about proteins and how they get targeted and moved into membranes and organelles. All right, so let's look at fundamentally how proteins are sorted in eukaryotes. So mammalian cells have approximately 10,000 different types of proteins. And so this is types, not just total proteins. So there's 100,000 plus different specific proteins within mammalian cells, especially in humans. Yeast have less, they're about 5,000, and that's kind of a model for some of these studies. So all nuclear DNA encoding mRNAs are translated in the cytoplasm, right? So we make mRNA, it goes to the cytoplasm, and that's where translation occurs. And so delivery of these newly synthesized proteins to their proper destinations occurs, there starts in the cytoplasm, and it occurs specifically during translation or soon after the translation process. So if mRNA does not have a signal to be excreted, which we'll get to in a minute, we go on this right pathway here. And so first we have the translation of the mRNA into a cytosolic protein. And if that protein's job is to remain in the cytosol, then that's it. So it gets translated and it's already in the right place, ta-da, and it's done. However, some proteins have to go to organelles or back to the nucleus or peroxisomes or things like that. So on these proteins, there will be a targeting sequence. So at the very end, whether it's C-terminal or N-terminal of this protein, there'll be a sequence that is meant to help traffic that protein to its proper destination. So in the case of you can see different targeting sequences will target them to mitochondria, if you're in plants, to the chloroplast, to peroxisome, and to the nucleus through these nuclear pores. Now, if this protein is to be excreted, then it goes down a different pathway. And so this is our pathway on the left here. And so the ribosome initiates this protein synthesis in the cytosol, just like it would be if it's a cytosolic protein. But as you can see here, we have an ER signal sequence, or endoplasmic reticulum sequence. And so this ER signal sequence directs the ribosome docking onto the rough endoplasmic reticulum import apparatus. So as this protein is being made, you can see that the ribosome is going to dock onto this rough ER apparatus. And then the nascent proteins are translocated into the ER or embedded in the ER membrane. So as you can see here, either A, we have our little blue ones here that are imported into the ER, or they are put in the membrane of the ER. Now, once that occurs, proteins in the ER membrane or in the lumen or inside can move via these transport vesicles to the Golgi apparatus. And then further sorting of these proteins to the plasma membrane, where the lysosomes occurs via vesicles. So you can see they're put into these little vesicles here that allow them to be transplanted into the membrane or into the lysosome. So let's talk here a little bit about the ER. So all eukaryotic cells have an endoplasmic reticulum organelle, and there are these convoluted tubes and kind of flattened sacs that have a large surface area membrane, and this is continuous with the membrane of the nucleus. The location of this is also the location of cellular lipid synthesis that we talked about previously. That was within the smooth ER, and it's the first step in the secretory pathway, secretion pathway, is within the rough ER and the attached ribosomes. And the ER is also the synthesis location of membrane proteins of the plasma membrane and the lysosome ER and Golgi apparatus or Golgi complex membranes. And it's the location of synthesis of soluble proteins that are secreted or function in the lumen of the ER or Golgi or the lysosomes. So in our figures here, we have a picture of the ER in pancreatic cells, and these cells are going to create proteins that are excreted. And you might think, when is a protein excreted and not within a cell or very near a cell? So in the case of the pancreas, we excrete enzymes into the digestive lumen or the digestive tract to digest food, right? And so all those digestive enzymes are proteins that need to be out of those cells. And so that's one example of moving a protein out of the cell. And then over on the left here, we have our kind of a close-up of the docking of these ribosomes that occurs during synthesis. And the ribosome that is floating free in the cytosol and the ribosome that is attached to the ribosome, those are identical. So there's not different ribosomes that attach to the membrane and different ones that float free. They're completely identical. And what actually targets those ribosomes to dock on the membrane is the sequence of the mRNA that is being translated. And so it's recruited to that membrane based on the polypeptide chain that targets to that ER membrane. So how did they figure out this targeting of proteins into the lumen of the ER and kind of determined what mechanism actually drives this? And so in this kind of a summary of an experiment or multiple experiments that they did to determine this. And so what they did is they took ribosomes and they put RNA with them and all the components needed, all the amino acids and everything needed for translation, and they allowed those ribosomes to transcribe the protein, or translate the protein into these complete sequences that do have the signaling sequences. And in this setup, there was no ER present, though, for the importing of those proteins into the lumen. So after the proteins are already created, then they added a microsome. A microsome is kind of a small version of the ER membrane. You can get these by kind of breaking up the ER membrane, and we talked about how these phospholipid bilayers naturally form these encased bubbles, and so that's essentially what it is. It's kind of like a proxy for the ER membrane. And once they added these after the proteins were already constructed, they looked inside those microsomes and they saw that there was no incorporation into the microsomes. So it wasn't this signal doesn't import after the fact that the protein's already made. Now, conversely, they did the same experiment, but they started with the microsomes present as well. So they put the ribosomes, the mRNA, all the amino acids and everything necessary to do translation, and the microsomes, and then they looked at the microsomes after this experiment. And what they found was that there was protein without that signaling sequence inside the microsomes. So what happens is that those microsomes recruit those ribosomes to their surface, and then the translation occurs where it drives the protein synthesis inside that organelle, or inside that microsome, excuse me, and then the targeting sequence gets trimmed off because you don't need it anymore once you're already there. And so this was kind of one of those experiments where they found how this import into these microsomes or into the ER lumen occurs. Let's look a little more in depth at this co-translational translocation. So in step 1 here, the N-terminal ER signal sequence, which is right here, SS, emerges from the ribosome first during this protein synthesis. So the targeting sequence is the first thing to be translated. And then in step 2, the signal recognition particles, or the SRP, which is right here, that comes in and binds to that signal sequence, and it arrests protein synthesis. So once it's bound right here, it pauses protein synthesis as it's bound to that signal sequence. In step 3, the SRP, the nascent polypeptide chain ribosome complex, so that's this whole ribosome developing polypeptide and SRP bound together, that binds to the SRP receptor in the ER membrane, so that's down here. So this whole thing folded up ribosome SRP polypeptide will bind to this protein that spans the transmembrane, or the ER membrane. And the interaction is strengthened by the binding of GTP to both the SRP, which is the darker green one here, and the binding protein down at the bottom here. In step 4, we have the transfer of this growing polypeptide chain to the translocon, which is this transmembrane protein complex here that is kind of a pore into the ER. And the GTP, will be hydrolyzed in both of those cases to GDP, giving off a phosphate group as well, and that unbinds those two as well. In step 5, the elongating polypeptide chain passes through that translocon channel into the ER. So as you can see here, it's moving on in and being transcribed into this lumen. The signal sequence is then cleaved by a single peptidase, peptidase being a enzyme that cuts peptides, and then this cleaved sequence signal here is rapidly degraded, so it's gotten rid of. Then in step 6, the growing polypeptide chain continues its extension through that translocon into the ER as we continue to finish this transcription of the mRNA. And then step 7 here, we have we get to the stop codon, and then in step 8 we have protein folding, and the translocon closes and the ribosome is released. So let's look a little bit at this translocon protein. So the role of this is to allow that peptide chain to be translated into the lumen of the ER. And so it also has to preserve and prevent other things from coming in. You don't want things to slip into this lumen while the peptide chain is growing through it. So it has kind of a twofold gating mechanism that is there to prevent other things from coming in. And so the first thing is, before there is any peptide chains coming through, there is this alpha helices plug that goes in and plugs the lumen side of the translocon protein and prevents things from kind of moving through that pore into the lumen. And secondly, when a peptide chain is moving through this translocon protein, there is this ring of isoleucines kind of at the waist, as they say, the very narrow part here of the pore, that forms this gasket and kind of keeps the channel sealed to this small sealed around that peptide chain and prevent small molecules from kind of slipping through with it. So it's a very narrowing of the pore that fits very tight to that peptide chain that prevents like sodium or other small ions and things like that of kind of slipping through as well. So it does a very good job of only letting those peptides through as opposed to allowing, you know, opening the floodgates, so to speak, and allowing a lot of stuff to get through. So there are 5 classes of these topological ER membrane proteins. And so let's look at these kind of specifically. So there are integral membrane proteins. These are classified by orientation within the membrane, the locations of the N and C termini, so those are the ends of that polypeptide chain, and the types of targeting signals that they contain. There are hydrophobic alpha helices segments that are embedded in the membrane bilayer. So those generally are the segments that are going to span the bilayer in most transmembrane proteins. I don't want to say always, because there's always exceptions to the rule within biology, but for the most part, alpha helices are what span these lipid bilayers. There's hydrophilic regions, and these are the regions that are exposed to the cytosol or to the lumen of the organelle, and they fold into various conformations outside the membrane. And so looking at type 4 proteins here, these are multiple transmembrane helices. And so as you can see, the same protein, the same amino acid chain, spans the membrane multiple times. And that's what makes it different from type 1, 2, and 3, as well as the tail-anchored proteins, is that this one goes across multiple times. And it's a G protein that has coupled receptors. And in our example here, we have these 7 alpha helices that go across. We have an N terminus on the exoplasmic side of the membrane. So that would be the bottom here. And then we have a C terminus on the cytoplasmic side. And then the other type 4 helices, they can have a different number of helices spanning it. So it's just generally multiple helices spanning the membrane is kind of indicative of type 4. So as you can see, there's the 5 different types here. We have the type 1, which is LDL receptor. We have type 2, which is a transferrin receptor, and they also have different, you can see growth hormone receptor for type 1, etc. But you can kind of see the different motifs here. So tail-anchored proteins are ones that only have the extracellular or extramembrane peptides on the cytosolic side. You can see that in type 1, we have the C-terminal domain on the cytosol side. Type 2, the C-terminal domain is instead on the exoplasmic side, etc. So those are some of the differences here between types 1, 2, 3, 4, and tail-anchored proteins. So I'm not going to go over everything specifically, but these are good things to know down here, hint, hint, for the exam. So now let's go through and look at how these are inserted into the membrane. We're going to go through each class here. So we'll start with type 1. These are single pass transmembrane proteins, and this is the type that have the N terminus in the ER lumen. And it's got a single transmembrane domain, and then the C terminus is in the cytosol. So as you can see here, we have the C terminus and we have the N terminus, and we have the C terminus in the cytosol. As you can see, it is the top here, and the lumen is the bottom. So how does this work? So in step 1, there's translocation initiation and SS cleavage by the same mechanism as for those soluble secretory proteins that we had talked about previously. So you can see that there's the binding to this translocon here protein. We have our ribosome that is bound to it. We have a signal peptidase here, and it is starting to translate from the ribosome through that membrane. Now, in step 2, we're just getting elongation here, right? So the transmembrane peptide is elongating. In step 3, the elongation continues until a hydrophobic stop transfer anchor sequence enters the translocon. So there's a specific sequence, the stop transfer anchor sequence, that says, okay, this is that transmembrane motif. And so once that gets made, we don't want that transmembrane part to go too far and go into the ER lumen, right? We want it to stay in the membrane. So this translocon protein will prevent this chain from extruding further into the lumen, because it's like this alpha helices here, it's supposed to be here, so we're not going to let it slide through anymore. So in step 4 then, the stop transfer anchor sequence moves laterally through a hydrophobic cleft between this translocon subunit, this purple, where's my cursor? Purple membrane thing here, and the phospholipid bilayer, and then the translocon closes. So it sneaks out, right? The chain does here. And now that we have anchored our alpha helices, we don't need this translocon protein anymore. So it closes, it's done. In step 5, the synthesis will continue, and the elongation chain loops into the cytosol through a small space between the ribosome and this translocon. You can see kind of right there. And then in step 6, the ribosomal subunits are released into the cytosol, and then that free protein end, the amino acid chain or the nascent protein, it will diffuse laterally into the ER membrane, and then we start to get post-translation modifications and protein folding and things like that. Okay, let's talk about type 2 and type 3 single pass transmembrane proteins. And so unlike our example of type 1, these proteins lack a signaling sequence. So if you look in the last slide, there is a signaling sequence on the N-terminus that is cleaved off, and that tells the protein kind of how to orient. But this is different in type 2 and type 3 in that there is no terminal signaling sequence. Instead, they have a single internal hydrophobic signal/anchor sequence. So that alpha helices that spans the membrane is both a signal and the anchor for this protein. So we'll start with type 1 here, and in this we have the SRP that binds to the internal signal anchor sequence that's not shown here, and then it interacts with a receptor on the membrane. So our previous steps, we're kind of jumping to the point where we're actually anchored in the membrane here. So look at our previous steps of how this anchoring occurs with the SRP, etc. It still occurs, but we're cutting to the chase, so to speak. So then the positively charged amino acids on the N-terminal side of the signal anchor sequence orient that peptide chain into the translocon with the N-terminal position in the cytosol. So as you can see here, the N-terminal is up in the cytosol, and we are creating our peptide chain, and then it goes through this signal anchor, or this back again through the translocon, and it will stop once the signal anchor sequence is recognized by the translocon here, right? I'm going to circle this blue because everything's red and it's kind of hard to see. So once that occurs, then that anchors in place, and the rest of the peptide as it's being translated will end up being kind of extruded into the ER lumen. And once we get to the end, to the C-terminus, the ribosome will break break off. It has nothing left to transcribe to translate, and we have our protein that spans the membrane. So in type 3 proteins, in part B over here, it is the same thing but flipped, right? So we start, instead of starting by translating or creating the protein peptide chain in the cytosol, we start by putting it into the lumen. So it's just backwards. And then eventually, once this anchor/signaling sequence is recognized by the translocon, what it does is it locks it in place, and we start to translate the rest of the polypeptide chain, the C-terminal side, in the cytosol. So as you can see, the only differences here between these two proteins, I'm going to circle this in purple, the C-terminal domain is in the lumen in type 2, and the N-terminal is in the cytosol. And then in type 3, the C-terminal is in the cytosol, and the N-terminal is in the lumen. So moving on to tail-anchored proteins. So these proteins have a transmembrane domain, but they really don't have anything on the lumen side. So they're anchored into the membrane, but then their N-terminus is just floating out, and the C-terminus really isn't in the lumen. And so how does this work? And so there's no N-terminal signal sequence, as I had said, and the hydrophobic C-terminus is where the transmembrane alpha helices is located. And this doesn't get inserted into the membrane until the protein is completely translated. So unlike our previous examples of type 1, 2, and 3, these are inserted after they're fully translated. And so the mechanism for this is there is a group of proteins, SGT2, GET4, and GET5, and they sequester this nascent protein C-terminal tail anchor and transfer it to this GET3 ATP. So we don't really show SGT2, GET4, and GET5, but what they do is once this protein has been translated, they safely take care of that hydrophobic region, right? Because the hydrophobic region doesn't want to be in cytosol because it wants to be buried away from any sort of water. And so they take care of that hydrophobic region and then help it get to this GET3 complex there and then be protected. Then in step 2, this GET3 ATP complex, it hydrolyzes ATP to help dock with GET1 and GET2, which are these transmembrane proteins. And during that hydrolysis, the hydrophobic tail of this tail-anchored protein is then transferred in between GET1 and GET2. And so GET1 and GET2, they kind of protect this hydrophobic tail and help get it into that membrane. So they kind of provide this channel, and because they're able to be interacted with by GET3, they can kind of change the conformation of GET1 and GET2 to kind of facilitate this insertion of that hydrophobic tail. And then at the end of step 3 here, GET3 releases, and it releases that ADP, which allows it to be recycled. So now that the ADP is gone, ATP can come in, and this enzyme is used over and over to insert these proteins. Once the release occurs, the protein here is released also by GET1 and GET2, and this protein then is anchored, and it no longer needs the help of any of the GET proteins or SGT2. It has been inserted. Okay, so as you can kind of glean from the last few slides, there are these topogenic sequences, so specific signal sequences that determine the orientation of the membrane protein, the ER membrane protein. And so this is kind of a summary, which is very nice, especially for trying to kind of decipher all this information in these different proteins. So type 1 proteins, they have that N-terminal sequence. So this is that sequence that originally targets them to the membrane, and that gets cleaved off. And then they have a single internal stop transfer anchor. So that's that alpha helices that anchors it to the membrane. In type 2, there is this single stop transfer anchor alpha helices, and also the orientation depends on this high density of positively charged amino acids that we didn't get into a lot, but this is on the N-terminal side of the stop anchor sequence. So in type 3, it's the same thing. We have this single SA internal sequence, and the high density of those positively charged amino acids are on the C-terminal side. And so, like I said, we didn't really get into the mechanism of that, but just know that this is a feature that just helps to distinguish type 2 and type 3 transmembrane proteins. So in type 4, there's no N-terminal signal sequence. The only one with an N-terminal signal sequence is type 1. The N-terminus ends up in the cytosol, or sorry, these are type 4A, so there's A and B. In this one, the N-terminus is in the cytosol, and there are these alternating kind of type 2, so I'm going to put that down here, SA, where there are the positively charged amino acids on the N-terminal side, right? And so as you can see, we have then alternating cytosol, lumen, cytosol, hydrophilic domains. And then in type 4B proteins, this is more similar to the type 3, but occurs multiple times, right? So this also goes over the membrane a bunch of times, but we also have these C-terminal amino acids that are positively charged. And so you can have N-terminals, but the majority of them are the C-terminal positively charged signaling sequences that are near that signal anchor sequence. So lastly, we're going to talk about these classes of these proteins, transmembrane proteins. So this one isn't really it's kind of a subgroup or an offshoot, because this doesn't have a transmembrane domain like the type 1, type 2, type 3, etc. So these are the GPI-anchored proteins, and we'll see why they're different. So bear with me here, because this is a word. So we have this amphipathic glycosylphosphatidylinositol. We're going to call that GPI. So don't worry about trying to memorize that. Glycosylphosphatidylinositol, GPI molecule. Okay, and this GPI molecule, they anchor covalently attached proteins in the membrane, and there is a membrane portion, a hydrophobic portion, as you can see here, and a polar portion, or a hydrophilic portion. So this GPI molecule, the hydrophobic portion is a fatty acyl chain, and the hydrophilic portion are carbohydrate residues and associated phosphate groups. So you can see that in part A here. And the length of the acyl chains and the carbohydrate moieties may vary depending on the species. So our example here, I believe, is in yeast, which is highly studied. And then if we were to look at how this anchoring occurs, this GPI anchor protein forms when protein is synthesized and inserted into the ER membrane, just like our type 1. So as you can see here, this part on the left-hand side really resembles that type 1. So that's why this isn't really its own class. It's just kind of a modification. So once that protein's inserted, there is this specific transamidase, so transamidase, that cleaves the precursor protein within this exoplasmic facing domain. As you can see, it cleaves it on the lumen side here. And then that occurs right near that, that stop transfer anchor point. And what it does then is it covalently links this new C-terminus, which it's created, which would be this part right here, and links that to the terminal amino group of this GPI anchor. And so this is, like I said, it's not really its own class, it's just kind of a modification to the type 1s where they get transferred to this GPI anchor as opposed to having their own alpha helices transmembrane domain. So it's just, like I said, more of a modification than an actual group. So once these proteins are in the membrane, there's modifications that can happen to them, such as the addition of oligosaccharides. So we're going to look at how the biosynthesis of these oligosaccharides occur. And so there is this N-linked oligosaccharide that is added to many proteins in the rough endoplasmic reticulum. And these oligosaccharide precursors contain 14 residues, and they are performed on a dolichol phosphate that's embedded into the membrane, which is here. So that's kind of our first precursor. And the dolichol phosphate is a hydrophobic polyisoprenoid lipid, which contains 75 to 95 carbon atoms. So that's what's in this membrane for our starting point here. So the way this mechanism works is in steps 1 through 3, we have 2N-acetylglucosamine and 5 mannose residues. So the N-acetylglucosamine are these red, as you can see here, and then the mannose are the blue. And these are added with high energy phosphatate linkages, and they're added one at a time on the ER membrane cytosolic face. So they're added on the outside of the ER membrane. Then in step 4 here, the 7 residue dialcohol pyrophosphatol intermediate is flipped. So as you can see, we were on the cytosol side, now we are on the ER lumen side, so the entire thing just kind of flips over to the ER luminal face. And then in steps 5 and 6 here, we have additions of sugar by some of these enzymes that are only present in the ER lumen. So sugar is transferred from this nucleotide precursor to a carrier dialcohol phosphate. And so as you can see here, we're adding more and more of these mannose sugars to the chain. And then this, you can also see this, we have this 3-UDP up here. So we have a glucose molecule that is attached to those dialcohol phosphates, and they flip to the inside. So we're importing sugar, in this case glucose, which is then added to the oligosaccharide as well. So we're adding mannose from the inside. Well, excuse me, mannose is also imported in a similar mechanism, which is added while on the inside, and we import glucose while on the inside to be added to this growing oligosaccharide chain. So now that that oligosaccharide chain is made, it can be applied to these proteins. And so these oligosaccharides promote protein folding, stability, adhesion between different proteins, and recognition for like trafficking and things of that nature. So continuing on this mechanism, so we have our oligosaccharide chain that is in the ER lumen here. we then have this long chain of polysaccharides that include mannose and glucose and acetylglucosamine. And so it would be glucose 3, mannose 9, glucose 2 glucosamine. Don't worry about that, just a big oligosaccharide because they can be different depending on the proteins. Then in step 1 here, I guess 1 or going to 2, the oligosaccharide is transferred to the growing nascent amino acid chain. So it's while this chain is being created in or translated in the ER lumen. In steps 2, 3, and 4 here, we have the removal of those glucose residues. So as you can see, they're starting to be to disappear here. However, in step 3A, there is sometimes that one of those glucoses gets added back because it can play a role in the correct folding of many of the proteins in the ER. So like I said, it depends on the protein. So sometimes they get all taken away and then one gets added back to be part of that oligosaccharide. In other cases, they're all taken away. And then in step 4, we remove one mannose residue and it is then done. It's attached. And at this point then, it can be transferred to the Golgi apparatus if we're going to secrete the protein and things of that nature. So there's another modification that happens to these proteins. We have protein disulfide isomerase, or PDI, and that helps to form these disulfide bonds between cytosine residues within this growing polypeptide chain. Oops, it's this one here. So we have this oxidized PDI that within this enzyme, there are two closely spaced cytosines that are easily interconverted between this reduced dithiol form and this oxidized disulfide form. And so what happens is this oxidized PDI will interact with this reduced substrate protein after it's been translated, and it will form this disulfide with one of the two cytosines. And by doing that, this is kind of a temporary holder here, it will remove that electron and then allow this second cytosine to form a connection, a disulfide bridge, between the two cytosines, which is more thermodynamically favorable. And then this PDI will accept the electrons and it will be reduced. And as you can see, we then have a disulfide bridge across these two cytosines, this oxidized substrate protein. And then this oxidized ERO1 will come in and it will reintroduce this disulfide bond in PDI and allow it to continue to keep doing this. So it recharges. It's an enzyme, so it's able to do the process over and over again. And so in addition, this PDI, during this process of generating this peptide amino acid chain, there is often the cytosines that will form double bonds just out of happenstance or chance. And so that might not be the proper conformation of the protein. So as you can see in their example here, we have this protein with incorrect disulfide bonds. Instead, they should be forming across and not with their closer neighbors. So this reduced PDI will come in and it will form these bonds with the one of these cytosines, and it will form this disulfide bond and act as this terminal electron receptor. So these numbers here are the movement of the electrons. So you can see that one comes over to reduce this bottom bridge, and then above it we have the reduction of this top kind of disulfide bridge, and then lastly we have the full reduction of both the cytosines and the PDI. And what we end with here is these two bridges in the proper conformation. And so these reactions will continue to go on and on and on until the protein is folded into the most thermodynamically stable conformation, and at that point it's kind of a finished product because the affinity for binding with PDI is reduced greatly, and so it kind of makes sure through trial and error that it gets to the most properly folded conformation. Okay, so let's look at how some of these proteins fold after they are incorporated into the ER lumen. And so in this example here we have hemagglutinin folding in assembly. And this requires chaperones and other ER proteins, including lectins, to facilitate this rapid folding and assembly of this protein as it's being synthesized into the ER lumen. And so in step 1 here, we have this transient BIP, which is a chaperone. Get my marker. And the chaperone binds to this certain regions of this nascent protein as it enters the ER, and it helps to prevent premature folding and incorrect conformation. As you can see, these may look familiar. These are those oligosaccharides that previously were added to the polypeptide chain. So then in step 1b, so we have a step 1a and step 1b here, calnexin and calreticulin are both lectins, and they bind to these certain oligosaccharides that have been added, as we talked about in the previous slides. And this helps prevents that premature folding into incorrect conformations as well. So like I said, when we were talking about those oligosaccharides, those are kind of signaling molecules too. And so calreticulin and calnexin bind to those to prevent this improper folding. So then in step 2 here, PDI catalyzes the formation of these 6 disulfide bonds per monomer that we had talked about, the disulfide bonds formed by PDI before. And as you can see, these little orange bridges there. And once it is properly folded, the monomer releases those chaperones. Beyond that, in step 3, this is just showing that our example here of hemagglutinin is a trimer. And so 3 of these monomers that we saw in step 2 here, they come together and form this trimer that is bound to the membrane and extends into the ER lumen. So what happens when there are excess unfolded proteins? And so there's this kind of checks and balances that occurs with unfolded proteins in the ER lumen that ensures that there's enough processing ability to fold everything correctly. And so the presence of unfolded proteins in this rough ER increases the transcription of genes that encode those chaperone and other folding catalysts. And so we're going to talk about this IRE1 protein, which is right here. So IRE1 will bind that BIP chaperone, and once those binding domains are fulfilled, everything's fine. That's kind of like the normal state. But if unfolded proteins start to accumulate, those chaperone proteins will, BIP1, will be released to go and take care of those unfolded proteins and help them to fold. And this is kind of a signal that, uh-oh, we need more IRE1 because we're starting to use all of our reserves. So then what happens is these IRE1s, because they are no longer bound to BIP, they will form dimers. And once they form a dimer, their cytosolic domain, so let me step back, these are transmembrane proteins, these IRE1 monomers, so they have a ER lumen side that binds BIP1, and they have a cytosolic side. So moving forward again, once they form dimers, their cytosolic side gains this endonuclease activity. So they're now able to cut mRNA. Now this is where it gets a little weird. So as I've said before, there's always exceptions in biology, right? And so in this case here, we have this HAC1 mRNA that's floating around the cytosol. And this HAC1 mRNA has an intron. And as we've talked about throughout this course, and you've probably learned in other courses, in eukaryotes, that's not supposed to be a thing, right? We're supposed to have processed all those introns and spliced them out before they get to the cytosol. Well, however, here we have a rare exception. And HAC1 has an intron. And this exonuclease activity, what it does is it splices out that intron when it senses it needs more BIP1. And after that, the two exons here of this HAC1, they splice together. And from there, they become a fully spliced, correctly processed HAC1 mRNA, which then gets translated into a protein because we're already in the cytosol. And that protein will then go and be imported into the nucleus, which isn't shown here, but I'll kind of draw one. And here's my double-stranded DNA. Oof, that was a little rough. And it will go into the nucleus and act as a transcription factor to specifically turn on chaperone genes. So then you can think of this further downstream. So then we're turning on chaperone genes that are mRNA that come out, and then they go to our ribosome, and at the ribosome they get turned, or well, they meet here on the ER membrane, and then put BIF proteins into the ER lumen to fulfill that need for more chaperones. So we talked about those end-linked oligosaccharides before and how they are also responsible for helping with folding, but they're also there to help with quality control. And so misfolded proteins are recognized by specific ER membrane proteins, and they're targeted for transport from that ER lumen into the cytosol to be degraded and then reused, or those components will be reused. So the mechanism for this is that calnexin and calreticulin, which we talked about previously, they bind, where's my marker here? They bind to the end-linked oligosaccharide in which that glucose was re-added. We talked about how glucose was completely taken off, and then one can be re-added. And they retain protein in the ER for folding chaperone activity until properly folded. So they're there to help and maintain that connection until properly folded. But with proteins that cannot fold properly, they stay in the ER lumen for longer amounts of time, right? Because they're not ready to be let go or secreted because they're not folded properly. And eventually they undergo mannose trimming by this ER alpha mannosidase, so it trims mannoses, and that forms this mannose 5,6 GLC NAC. And when that occurs, this OS9, which we have right here, that then is able to bind. So as you can see, this the calnexin and calreticulin bind a specific pattern of sugars. So we have mannose, mannose, glucose. And after this trimming by this glucosidase and then this alpha mannosidase, we have mannose, 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 and they're in a different conformation. So that signals to OS9 that this is misfolded, and we send that protein out into the out of the endoplasmic reticulum back into the cytosol, where ubiquitination, which I believe we've talked about, and degradation by this cytosolic proteasomes occurs. And so then we'll recycle that protein into amino acids, which then can be used to create more proteins. So we've talked a little bit about the targeting sequences that some different proteins have, or mRNA that will generate proteins in the case of the ER. And so this table, I'm not going to read it to you because you can read it yourself, but know that there are different targets sequences for different organelles. They occur in different locations. The endoplasmic reticulum has an N-terminus target. The mitochondria and the chloroplast in plants have N-terminus, but the peroxisome mostly has a C-terminus target. it depends on what organelle they're going to, whether or not they are removed or not, these sequences during the process of folding, and there's different natures of the sequence. But like I said, I'll leave this for you guys to look at and kind of decipher. I'm not going to read it to you, and it might be something good to know for the exam. Hint, hint. Okay. So now let's look at another organelle and see how proteins are imported into the mitochondrial matrix. And so our first step here is that, pre-step, is that precursor protein synthesis occurs on the cytosolic ribosome. So in the cytosol, proteins are being made just like normal, and then we go to the import steps. So in step 1, there's the amino acid chain is maintained in an unfolded state by bound chaperones such as HSP70, which is heat shock protein 70. In step 2, there's this matrix targeting sequence that binds to the outer membrane import receptor, which is this green guy down here. And this is near this rare contact site with the inner membrane. And so once that occurs, this MTS, so matrix targeting sequence, is inserted into the outer membrane translocon. So as you can see, similar to our previous examples, this purple-ish, blue, purple, depending on your computer screen probably, the import receptor just kind of puts it into the translocon, which is that transmembrane channel for the amino acid sequence to move inside. In step 4, the translocating protein moves through and inserts into the inner membrane translocon. So remember, this mitochondria, kind of like the nucleus, has 2 membranes, and there's an inner membrane space. So we have our outer membrane, and we have our inner membrane, and then we have the mitochondrial matrix at the bottom. And so in step 4 here, this protein moves through the outer membrane and inserts into the inner membrane translocon, which we need 2 of them to get through 2 membranes, right? In step 5, the protein moves through the inner membrane. It binds to HSP70, and that helps drive the import into the matrix. So we have another heat shock protein inside here and helps to kind of pull it through into the inner matrix. And this MTS, the matrix or mitochondrial targeting sequence, is then cleaved off because we don't need it. It's just the targeting sequence, and we don't really want to incorporate it into the protein. In step 6 here, we have this heat shock protein 70 hydrolyzes and releases that imported protein. So the hydrolysis of ATP to ADP causes the release of that heat shock protein that was there to help get that protein in, and it allows it to be kind of floating freely in the internal matrix there. And then the protein there folds into its mature active conformation within the intercellular matrix. So there are 3 distinct pathways that get us from the cytosol into the mitochondria. So we're going to briefly look at these. And so these different pathways require different targeting sequences, or multiple targeting sequence. But all of them require TOM40, this outer membrane channel to import into the mitochondria. So in path A, we have COXVA. Let's see, I don't see it labeled on here, so it's COXVA, COXVA. And this is a matrix targeting sequence and hydrophobic stop transfer anchor sequence. We talked about those anchor sequences where they kind of get stuck in the membrane, and that's what COXVA is. So this N-terminal matrix targeting sequence that we have right up here, that is recognized by TOM20/22, this recognition complex on the outer membrane. So as you can see here, it's recognized and then funneled into TOM40. And this hydrophobic stop transfer anchor sequence blocks the transfer through the inner membrane. So once it goes through TOM40, it will continue in through TIM44. So TOM and TIM, OM for TOM stands for outer membrane, IM stands for inner membrane. So TOM and TIM are kind of the same thing, but just on different membranes. So this matrix targeting sequence will be recognized by TIM44 here. And when that occurs, this blocks transfer through TIM, and it releases TIM into the inner membrane. So that stop transfer is recognized there, and it says, okay, this is as far as you go, stop and release. Okay, in path B here, we have this OXA1 targeting sequence, and this is in the middle of our protein. And this is similarly imported by TOM9 and recognized by TOM20 and 22 and imported into the inner membrane space. But where it differs here is that once going through TIM 23, 17, 44 complex here, the complex recognizes that OXA1 targeting site, and this inserts the polypeptides with the help of OXA1. So the OXA1 targeting site obviously interacts with OXA1, and it inserts it into the inner membrane there. And as you can see, we have two of these alpha helice transmembrane domains. So with the help of heat shock protein 70, it folds this protein over, so we have a single protein with two transmembrane domains. And so we get this like horseshoe shape going across there. And then path C we know a little bit less about compared to path A and path B, but we'll talk about it here. So these are proteins that don't have a matrix targeting sequence. Instead, they still get imported through by TOM40, but instead of with 22 and 20, they use TOM22 and TOM70. So TOM70 helps to recognize non-targeting sequence mitochondrial proteins. And then once we get into the inner membrane space, TIM9 and TIM10 help to kind of further the movement into the inner membrane, where TIM22 and TIM54 will help to fold this protein and to assemble it with its multiple transmembrane alpha helices across this inner membrane. And so, like I said, this is a little bit less understood than the other two pathways at the moment, but it uses, as you can see, TOM40, but then completely different TOM and TIM transmembrane proteins or channels to get to that inner membrane. Okay, so we've talked about how we get to the mitochondrial matrix, the three pathways, but what about the inner membrane space? So there are proteins that hang out in this inner membrane space of the mitochondria as well. And so we have two pathways. The first is the cytochrome B2 pathway. And the way that this works is it's similar to those previous pathways where we have TOM40 and then we still have TOM20 and TOM22 that use that matrix targeting sequence up at the top here. And they get it to the TIM44, 23, 17 complex, where once across there is this inner membrane, I'm going to change my colors here, inner membrane targeting sequence. That inner membrane targeting sequence gets caught inside the TIM44, TIM23, TIM17 complex, and it stops there. And then what happens is this protease comes in, and what this protease does is it trims the polypeptide signal on the inner membrane side. So it cuts right there, or on this one it cuts right here, and allows the tail end of that polypeptide to kind of get cut off and drift away into the inner membrane space. Then the inner membrane domain and the targeting sequence, they get the protease activity comes in and breaks those apart, so we don't need those anymore. So they're just there kind of as two different targeting signals. One to get to the mitochondrial matrix, and then one that's kind of our stop site, the inner membrane space targeting sequence, to prevent too much from going into the mitochondrial matrix and allowing this polypeptide to be cleaved and then float away into the inner membrane space. The last way we have is part B here. In part B, there is, again, no matrix targeting sequence, and TIM9 and TIM10 help to take this polypeptide chain and move it to TOM40, our always active outer membrane transmembrane channel. And once inside, since there's no targeting domain, there's no way to move that protein across TIM44, TIM23, and TIM17 into the mitochondrial matrix, and instead there's these two enzymes, MIA40 and ERV1. There we go. And what they do is help to chaperone and fold that protein and form the disulfide bonds responsible for a functional protein. All right, so we're going to talk about the nucleus here lastly in this lecture. So we have the nuclear pore complex at different levels of resolution within this figure. And the nuclear envelope is a two-membrane system just like the mitochondria, and this separates the nucleus from the cytoplasm. Although the outer membrane is contiguous with the ER membrane, and these nuclear pores span both membranes for transport between the cytoplasm and the nucleus. So this is different than the mitochondria, which had pores that went from the cytosol to the inner membrane, and then ones from the inner membrane to the mitochondrial matrix. So in this figure here, we have just some EM figures of these pore complexes in Xenopus eggs. Take for it what you will. So they're very difficult to comprehend, I think, compared to the figures. But looking at figure B here on the right-hand side, we have the nuclear pore complex model and the major structural features that are formed by membrane nucleoporins, structural nucleoporins, and FG nucleoporins. And so as you can see here, we have this nuclear envelope here. We have the outer membrane and the inner membrane, and as said before, the pores are between both membranes. So it's not like the mitochondrial, where we had kind of these single TIM and TOM40 that were allowing things in. Here we have pores that go between both envelopes here. And so down below, we can see this kind of the scaffold structure on this figure down here. And there are 16 copies of this Y complex that they're kind of indicating here. And these are what kind of form that pore, or if you look from the top view, they kind of form the ring to allow that pore to exist. And then lastly, we have these FG nucleoporins on figure D, where we have this kind of extended structure. And these repeat sequences that help to form, as you can see. So to orient you, these are inside here, right? So these are kind of the scaffolding that help to keep that pore open. They would be also, this is kind of a zoomed in sequence here that you can, or image here, that you can see that they would be kind of throughout this, that provide that scaffolding to help keep this open, and it's interspersed with hydrophobic and hydrophilic regions. So we know how the pores work. How do things get into the nucleus now? So we have this kind of cargo transporter system here to import. And so in the cytoplasm, there is this aptly named protein called importin, which is there to import proteins into the nucleus. And this binds a cargo protein. So this can be any protein that has this NLS motif that is able to bind to important and be brought in to the nucleus. So this can be transcription factors and things of that nature. And so this important cargo complex diffuses through the nuclear pore complex and interacts and kind of goes through those FG nucleoporins that were kind of the scaffold of this pore. And then it gets into the nucleus at this point. And within the nucleus, there is this enzyme called RAN, R-A-N, GDP, which they didn't label here for some reason. But RAN GDP is activated by guanine exchange factor, or GEF. And through that process, it becomes RAN GTP. And then RAN GTP is able to take up this bond with our importin. And when it does that, it releases our imported protein. So it comes in and it says, get out of the way, imported protein. I need to bond with importin. And when that happens, the imported protein is now where it's supposed to be, in the nucleus. But that's not the end of our story, because we have to continue. A lot of these import mechanisms we've seen are cyclical, right? And so now importin and this RAN GTP go back through a nuclear pore, interacting with the FG nucleoporins to get through. And on the outside here, this RAN GTP is hydrolyzed by this GAP protein. And what that does is it releases this phosphate group and it converts RAN back to a GDP. And we're not showing there's also a mechanism to bring RAN GDP in to the nucleus, which isn't shown here. But important now is free to continue the cycle, and RAN GDP will go into the nucleus, be converted again, etc. So there's kind of two cycles, but we're looking at only one at this point. So sometimes we also have proteins that need to go from the nucleus out of the cytoplasm. So as opposed to being imported, they're being exported. So we're going to look at how this works. So there is two pathways. There's a RAN-dependent and a RAN-independent export pathway. And so in the RAN-dependent mechanism, which is on our top here, we have this nuclear export signal, which is this we call an NES, and it's a leucine-rich region. And exportin 1, which is similar, it looks almost like importin in the last slide, but it's this red middle complex, protein complex there. And it binds cooperatively to form a complex with this NES protein, and again with RAN GTP, which seems to be important for both import and export. And once this complex is formed, it diffuses through that nuclear pore complex and interacting with those FG nucleoporins, just as we saw, similar at least to the importin complex or method. Once in the cytoplasm, RAN GAP is associated with the nuclear pore complex cytoplasmic filaments, and this stimulates that RAN GTP hydrolysis. This GAP here, it senses those filaments and then starts this hydrolysis process. And that hydrolysis process causes a conformational change and releases that cargo into the cytosol. So GAP is there to release the cargo. Once that occurs, just like the last system, this is a cycle, so it goes round and round, and so we need to move the exportin and the RAN GTP back into the nucleus for the next round of export. And so exportin 1 and RAN GTP are transported back through this nuclear pore complex, and once there, RAN, with the help of GEF, just like in our last example of importin, is converted back to RAN GTP, which will allow this export again. So the RAN-independent mechanism takes a step back. It's not exporting proteins, but mRNA to make proteins, right? And so what we have is in the nucleoplasm, or in the nucleus here, we have our capped and poly-A mRNA, and this heterodimeric complex, so two half proteins make a whole complex, NXFT and NXT1, or sorry, NXF1 and NXT1, they bind as a dimer to this mRNA and help to navigate it through the nuclear pore complex by interacting with those FG nucleoporins. So that's what guides them through this pore complex. And in the cytoplasm, there's an RNA helicase that we call, where is it on here? DBP5, right down there. DBP5 is there to use ATP energy, so ATP converted to ADP, to remove NXF1 and NXT1 from the mRNA. And then it's free to be, you can see it's going up to be translated here into a protein. But NXT1 and NXF1 are then re-imported, just like all of our other mechanisms here, into the nucleus, or the nucleoplasm, through those the nucleopore complexes and interacting with those FG nucleoporins, and they're able to export more mRNA. So that is all for this chapter. We're at an hour and 15 minutes. This went long for 31 slides. So I will see, we're getting towards the end here. Hang tight, and I will see you in the next chapter.